So the next topic is Agrobacterium tumefaciens, TI plasmid, or known as PTI. In last lecture we have discussed characteristics or properties of vector, choice of vector, and few plasmid vectors PUC18, PUC19 and PBR322. Now today we are going to discuss PTI, that is plasmid extracted from tumor induced plants. So these Agrobacterium tumefaciens, these are gram negative organisms found in soil. These organisms, they infect plants, they enter in plants through their roots and cause crown gall disease. Okay, crown gall disease. They infect at root crown or just below the soil line. They can survive independent of plant host in the soil. They infect plant through the breaks or wounds of the plants. Wounds of the plants, they secrete certain phenolic compounds which attract these agrobacterium towards the wounded plant, and then agrobacterium enters their plasmid through this wounded tissues. Common disease of woody shrubs, herbaceous plants, particularly problematic with many members of the rose family. This crown gall disease is commonly found in woody shrubs, herbaceous plants and rose family plants. Galls, that is the tumors, are spherical wart-like structures similar to the tumors found in plants. Okay, crown gall disease. Agrobacterium tumefaciens are responsible for this crown gall disease. So this disease occurs mainly by the presence of plasmids in this Agrobacterium tumefacien organism. This plasmid is known as TI plasmid. TI stands for tumor inducing plasmid. This plasmid consists the genes known as virulent genes. Okay, these genes are transferred to the plant cell, and then these secrete certain compounds which are responsible for inducing crown gall or tumors. Okay, the virulent strains of Agrobacterium tumefaciens contain 200 kb size TI plasmid, tumor inducing TI plasmid. Bacteria transfer a portion of this plasmid into a plant DNA or plant cell's nucleus. tDNA, it stably integrates this tDNA once transferred to the plant cell, enters into the plant nucleus. It stably integrates into the plant genome by recombination, express genes that result in physiological changes of the plant cells, and increases synthesis of plant growth hormones like auxins and cytokinins, and therefore increase in auxin cytokinin rate then increases the plant growth rate, and thereby plant cells divide and redivide and forms a tumor or gall. Now what are opines? Within this tumor tissues, the synthesis of unusual amino acids directed by the genes encoded on integrated plasmid. This Ti plasmid, which get transferred from Agrobacterium to the plant nucleus, it consists genes for unusual amino acids. Those unusual amino acids comes under the category of opines. Okay, opine is a group of unusual amino acid. These opine genes are present on Ti plasmid, which is transferred from Agrobacterium to the plant cell. The type of opine produced is specified by a bacterial tDNA. tDNA is the transfer DNA present onto the Ti plasmid, which is transferred into the plant nucleus and combines with the plant cell's DNA. Opines are used by bacteria as a source of carbon for their growth. Opine catabolism within the bacteria is mediated by the genes encoded on this Ti plasmid. So this is overall infection process of Agrobacterium. We'll discuss one by one. So this is plant tissue. These you can see, these are the plant cells with the cell wall. Now see here, number 1. This is cell wall and cell membrane of the plant is broken. So this is wounded plant. This produce phenolic compound known as acetosyringon. This acetosyringon attracts Agrobacterium tumefaciens towards it and activate virulence genes present on this Ti plasmid, present in Agrobacterium. This is Agrobacterium. Okay, this is bacteria which comes near close proximity or get attracted towards this wounded tissue, because this wounded tissue produce acetosyringon. This acetosyringon then activates vir genes. Vir stands for virulence. Genes responsible for virulence. So this vir protein synthesize single-stranded DNA. Okay, this DNA is a part of this Ti plasmid known as tDNA, which is transferred. Only the single-stranded DNA is transferred into the plant cell. Then from plant's cytoplasm, it enters into the plant nucleus. See, this is nucleus and these are pores present in nuclear membrane. Through this pore, this DNA get transferred into the plant's nucleus. This DNA, while it is entering, this is duplicated, that is converted to double-stranded form, and then integrates into the plant DNA, okay, which is present inside this nucleus. Now this tDNA, once it integrates, it increases the synthesis of auxins, cytokines and opines. Okay, opines. These opines, auxin and cytokines, promote tumor formation. Since these are plant growth hormones, if their production rate increases, they allow cells to grow, divide and redivide continuously. Okay, so this forms a tumor in Agrobacterium infected plant cells, and these opines which are synthesized, this helps bacteria to grow. These are used by bacterial cells as their carbon source, which are present near the roots of the plants. So this opine increases the growth rate of organism, and auxins and cytokinins increases plant cell growth and forms a crown gall or a tumor. Okay, so this is overall infection process. See, this is the tumor which is formed because of agrobacterium. So this is Ti plasmid. Size is 200 kilo base pair. It has its own origin of replication gene, tDNA, which is transfer DNA, virulence genes, and host specificity region, okay, which allow this tDNA to transfer to the specific host cell, that is plant cell. These virulence gene, they are 6 to 7 virulence gene. They are activated one by one because of acetylcysteine, and this virulent gene, they make transfer of this tDNA. They activate this tDNA cutting from this site and transfer into the plant cell, as well as recombination of tDNA into the plant genome. So this PTI is artificially formed vector from Ti plasmid. Natural Ti plasmids are used for preparation of this artificial vector, so that the genes can be transferred from bacteria to the plant cells. Okay, so this is the tDNA. This is also modified. This is TRA region. TRA region is for transfer, required for bacterial conjugation. These are the genes for opine catabolism, which are needed for bacterial growth, agrobacterium's growth. These are vir genes, which are needed for transfer of this tDNA. Unnecessary segments are removed, and instead extra segments can be attached. tDNA size is 23 kilo base pair, and it has borders at its left side and right side, known as left border and right border. The Ti plasmids can be classified on the basis of opines produced. One is octopine and another one is nopalin. Octopine and nopalin, these are two types of opines, unusual amino acids. Okay. Nopalin plasmids carry genes for synthesizing nopalin in the plant and for utilization in bacteria. Tumors can differentiate into shooty masses. Octopine plasmids carry genes required to synthesize octopine in the plants and catabolism of bacteria. The tumors do not differentiate but remain as callus tissue. These nopalins, they form number of tumors because this infection spreads, and this octopine plasmids, they do not spread their infection. It is localized infection, so tumor do not differentiate but remain as a single bulky mass known as callus. Agrobacterium tumefaciens chromosomal genes are CHVA, B, PSCA. See, you need not to go in so much of details. tDNA encodes the genes for opine synthesis and for tumor production. Vir region is present on PTI but not in the tDNA. These genes, they prepare tDNA for its transfer. It helps in excision of tDNA and transfer of tDNA into the plant cell. OCC genes are opine catabolism genes carried on the PTI and allow the bacterium to utilize opines as nutrients. So this, instead of tDNA is modified by removing auxin cytokinin genes and adding gene of interest. Okay. So this is mini TI, which has polylinker gene. This is left border of tDNA LB. This is right border of tDNA. Here in this middle part where tDNA is present, tDNA is deleted. Okay, and instead selectable marker gene, polylinker genes are attached. Bomb, these are Ori and bomb. Bomb is basis of mobilization required for mobilizing this region or tDNA to the plant cell. But here we are removing tDNA. So whatever gene you are attaching here in this polylinker site, what is polylinker site? It consists different restriction sites. Okay, sites for different restriction enzymes. So you can cut using any one enzyme, attach your gene of interest. This basis of mobilization gene will help your gene of interest to get transferred to the plant cells. This is how modified TI plasmids are prepared. See here, this plasmid consists kanamycin resistance and TRA region, PRK2031 plasmid. Then this is transferred from donor to the recipient. Okay. Now this plasmid has BOM, basis of mobilization site, and this is then modified. This kanamycin resistance is transferred from this plasmid to this, and then this is again modified by deleting tDNA. The oncogenes, that is tumor inducing genes in tDNA regions, are replaced by marker gene. It is also known as disarming of plasmid. Disarmed plasmid in the sense oncogenes which are responsible for forming tumor, that is the genes for synthesis of auxin and cytokinin, they are removed, and instead marker genes are added, selectable marker genes are added. Disarmed vectors do not produce tumors and can be used to regenerate normal plants containing foreign gene. In binary vector, the vir genes are required for mobilization and transfer to the plant reside on modified PTI. This is left right border kanamycin resistance, this is polylinker gene, origin of replication, and this one is basis of mobilization or TRA gene. Okay, this consists left border sequences selectable marker, that is kanamycin, this is not kanamycin, kanamycin resistance, and a polylinker for insertion of foreign gene. Then now this one consists, modified TI plasmid consists only vir region, origin of this plasmid, origin of replication of this plasmid, and deleted tDNA. Okay, So these two must be transferred to one organism known as binary vector, because this one mini Ti does not have Vir regions which are necessary for transfer of this tDNA segment, which is deleted from this site, but new DNA, foreign DNA is added here. Okay, so this was about tDNA vectors. Ti plasmid, PTI. So next vector is lambda phage vector, bacteriophage lambda cloning vectors. Now as we all know, bacteriophages, these are the smallest organism. This is the virus which infect specific host. It is a type of virus which infects specific host. The bacteriophage is the phage virus which infect E. coli bacteria. Okay, here we are considering example of lambda phage. Lambda is a type of bacteriophage infect E. coli. Okay, that is the host of lambda phage is E. coli. So this is the lambda phage. This lambda phage inserts its DNA into the host cell E. coli. Inside the host cell, bacteriophage replicates its own DNA, forms its own proteins, and forms heads and tails. Okay, then viruses are assembled and they are released out. Now this is the map of lambda phage genome. It consists head region genes, tail genes. These genes encode head protein, tail protein. This is non-essential segment, and these are replication genes. So these non-essential segment, these can be replaced with the gene of insert. Okay, this non-essential segment is known as stuffer fragment. Okay, stuffer fragment. This can be removed and your gene of interest can be ligated here in place of these non-essential genes. So the foreign DNA is ligated to left and right arms of lambda, then transfected into the E. coli cell as naked DNA or by packing in vitro. This foreign DNA, see, now this DNA is recombinant DNA because some phage genes are removed which are non-essential, which are not necessary for phage infection and replication, and your gene of interest is attached here, which is shown here with the red color. So this phage which consists recombinant DNA is recombinant phage. Okay, then these phages are allowed to infect to the E. coli cell and then allowed to release progeny phages after their growth. This you have to study, lytic and lysogenic cycle. I am not going to explain these basic things. So this is bacteriophage lambda genome. These lysogenic genes are non-essential, so this whole segment is removed. See, this whole segment is removed, and here new insert can be attached, which is of size 20 kb. What genes are required? Only replication, lysis, cos sites and head tail proteins. Okay, these are only required for phage assembly. Here you can attach your gene of interest. See, like this. Okay, this is non-essential segment is cut with the echo R1. This will generate segment sticky ends like this. Your gene of interest is attached here in between, like this. This is known as concatemer. When viral genome packaging is done, several viral genomes. See, viral genome replicates like plasmid, and during packaging, it replicates like rolling circle model. So this one end of the plasmid is pulled out like this, which consists multiple genomes, and each genome of adequate size is fitted into the phage head by cutting at specific locations known as pack sites or cos sites. From one pack site to the another pack site, this much DNA is accommodated in one phage head. Then from first pack site to the next pack site, phage genome is accommodated in or transferred in second phage head. Likewise, this many genomes of phages, when they are attached together, transferred, ready for transfer, they are known as concatemeric DNA. See here, the phage genome is inserted into phage head and cut at the cos site. Then from here to here is another cos site. This will be packed in another phage head. ATP is required for this process. Okay, once the specific amount of phage DNA is transferred, the head is sealed, middle piece is attached, tail fibers and tail fins are attached, and complete viruses are assembled. See, the same things are given for your understanding. This is gene of interest. This is stuffer fragment, which is not required. This is left and right border of the phage DNA. This stuffer fragment is removed and your gene of interest is attached here like this. Okay, then in vitro packaging is carried out and phages are allowed to release from the E. coli cell. So here you can clearly see these two ends are cos sites. This is non-essential segment which is removed, and this red color foreign DNA is attached here. The phage head is packed with the phage DNA from one cos site to the other cos site. These cos sites, these are complementary with one another. So inside the host, when this DNA is transferred into the E. coli, these can form a circular DNA since these two ends are complementary to one another. Once this phage genome is packed in one phage head, different regions of phages are attached. So there are two types of phage vectors. One is insertional vector, another one is replacement vector. Insertional vector contains only one unique site for restriction and large portion of non-essential region is deleted and two arms are ligated together. Replacement vector consists two sites of restriction enzymes. This larger segment is deleted major stuffer fragment is deleted, gene of interest is attached in between here. Lambda GT10, lambda GT11 are examples of insertional vector, and lambda EMBL4 is example of replacement vector. See here, only the difference is this consists only one restriction site, this consists two restriction sites. Okay, selection of recombinants is carried out on the basis of plaques. Now the viruses, you all know, when they infect any cell, animal or plants, animal or viruses, they form clear areas onto the nutrient medium. These are known as plaques. Certain phages form turbid plaques. Recombinant phages form clear plaques. Some have promoter, operator and lag Z gene. They produce blue colored plaques, while recombinants produce white colored plaques. Certain phages cannot infect bacteria which have P2 phage. They are spy plus, that is sensitive to P2 prophage inhibition, and while recombinants are insensitive to P2 prophage inhibition. Okay, so this was about